recorded. All right, the record reflects we are back in session. Parties and counsel are present. All members of our jury and our alternates are present, and you may continue your examination as proceed.
Thank you, Your Honor. So, Dr. Perlin, just a question. How do computers think in relation to probability calculation?
Systems like TrueAllele use something called Bayesian Reasoning. This is an idea from math that's over 250 years old. It has the concept that a thinking machine, whether a person or a computer, starts off with some initial beliefs, like what's the chance of a genotype in the population that could be based on scientific data. And then evidence is presented, and based on the data in the evidence, the belief changes to a new probability distribution. That is, the beliefs are realigned based on evidence. That's the basis of Bayesian reasoning. And Bayes wrote a formula down, it's published in 1762, I think, that says you can write down the probability of what you believe before you see data. You can multiply that by how well an explanation or an outcome or a hypothesis explains the data. Take those two numbers, and what you end up with at the end is what's the probability of that outcome, like a genotype value, based on the data. And what was interesting is no one had thought of that before. So the idea that what we really want to know is what's the probability of these genotypes, you can't get there without Bayes' theorem. You have to start with what you believed before, what the data is telling you, and then the probability of what you want to know, the probability of this value, of this assertion or this genotype, based on the data, can be calculated. So it's a way of updating our beliefs through data and math.
Now, in using TrueAllele, when it's used in criminal cases, what percentage is done for like prosecution versus defense?
At a crime lab that's using TrueAllele, it's probably almost all done for prosecutors. In the work we do at Cybergenetics, I'd say 90% of our work is with prosecution, and the other 10% is for defense, post-conviction, or maybe family testing for paternity.
And when you testify in cases regarding prosecution, it's usually to identify, I assume, the perpetrator?
Usually there are some cases where the lawyer, typically a prosecutor, from your question, is interested in knowing who's not on some items as well as who is on some items. But most of the time, the question is something like, did this suspect touch this gun? Or did they leave their DNA in some place they said they weren't? That's the usual question that a prosecutor would have.
And you heard the testimony of Mrs. Pujols about how cybergenetics was involved in this matter, correct?
Yes.
And there's previously testimony that Bodie Cellmark, or now Bodie Technology, developed the electronic data that was forwarded to cybergenetics. Did you have some part in the analysis of that data at cybergenetics?
I was consulting on that. I mean, I wrote the protocols and I wrote the program, but I remember this was a bit of a rush in early January before I was leaving town. So there was a lot of discussion in the team about what was needed and how to get everything done in time.
And were you involved in preparing any materials in this case to help explain to the jury what cybergenetics did with the materials from Bodie Cellmark?
Yes, there's a PowerPoint presentation. We have a protocol for preparing that as well. It was initially drafted, I think, by Beatrice, and then it went through several rounds of revision in discussion with you and your group. And then I made changes to it and put it into its final form. And
the final, well, let me ask you Are you ready to do that presentation, sir?
Yes.
Your court permission, can the witness come down to the well,
Sure.
using the microphone and the lectern to make his presentation? And for the record, we submitted a paper copy of this presentation as a court exhibit with judicial assistance.
It's the court exhibit at the end. Mary, what do you want it next in order?
Yeah, next.
So that would be 10 o'clock.
Okay.
Do you have a question, or can I just start?
You can explain to the jury what you did and the findings you've had. You have to turn the microphone on. Sorry. Yes.
Can you hear me? Okay. So this is describing the computer interpretation of the quantitative DNA evidence that we examined at cybergenetics in this case. I'll begin with some just background to explain what genotypes are and what it is we're actually looking for and how the computer calculates DNA match statistics. So the human body has trillions of cells that contain a nucleus. And inside that nucleus is our DNA. We inherit a complete copy of the DNA from our mother and another complete copy from our father. Both copies are inside that nucleus of most of our cells. And what that copy is 3 billion letters. It's text written in a DNA alphabet comprised of 3 chemical letters. It's not like on a piece of paper, it's chemicals that are together, but it's treated like text. And those letters are A, C, G, and T for the different base chemicals that are used. Those 3 billion letters of text are packaged into 23 chromosomes. And you have 23 pairs of chromosomes, there's one from each parent. This is what a chromosome looks like. You have seen pictures of them, and if you unravel the text, there's just a sequence of letters like G, G, A, C, T, A, C, whatever the text happens to be, and a location of interest. Whether it's the gene or the sort of random markers used in genetics and forensics, is called a locus. And so the loci, which is plural for locus, that scientists cared about in genetics in the 1990s when we were developing our computer interpretation methods, and are still used in forensics and have been for 20 years, are called short tandem repeats. And the reason for that is that there's a word that's usually short, typically 4 DNA letters long, like TGAC, and it's repeated in tandem. So when you have a word that's short, repeated in tandem, scientists will come up with a very descriptive name like short tandem repeats, abbreviated STR. There are about 100,000 of these STRs that are scattered through the 23 volumes of our genetic encyclopedia. Again, this is metaphorical. If you look in a cell, you don't see 2 complete copies of the World Book Encyclopedia. Just a metaphor, each volume corresponding to a chromosome. And out of those 100,000 locations, forensic scientists around the world have settled on maybe 25 to 50 particular paragraphs that are a page of a volume, that is a particular location on a particular chromosome. And they all have the same basic characteristics. There's text, and in almost all people, the text from the beginning to the end is the same. And the only variation, in most cases, is the number of times a middle word is repeated. So if you were to, I won't read it in English, but this is an English version. It's a popular poem. If you have a local baseball team, right, you recognize this. So there's the concept of this is that if all the paragraphs had the same number of words, there'd be no differentiation and you couldn't use it to distinguish between people. So scientists select certain loci where there may be 15 different variants. There might be 5 repeats, or 7 repeats, or 18 repeats. There's a lot of variation across different people. And what the scientists were doing back in the 1990s was trying to not sequence this sentence, this paragraph, to not work out what every letter was, but to work out what the genetic variation was just by the length of the paragraph. And the way they did this was like this. Suppose there's a certain length to this paragraph, and that length, if the first part is fixed, doesn't change, and after the repeated unit, root, that second part doesn't change, then just by measuring how many letters there are in the paragraph, you can figure out how many times the word was repeated. Subtract off the length at the beginning, subtract off the length at the end, take what's ever left, divide by 4, and you can measure the length to figure out that there are 10 repeated words. And if somebody had 11 repeated words, the paragraph would be 4 letters longer. So just by measuring the length of the paragraph, scientists could figure out which variant, which is called an allele, people had. And so this was based on length. And you'll be seeing different lengths that are corresponding to the different alleles. The more times the word is repeated, the longer the paragraph is, and that becomes the different allele. This allele is 10 because the word's repeated 10 times. If it was repeated 11 times, it would be called 11. So that's what the scientists are doing. They've selected different locations. They're measuring length. They're going to ask how much of each of these variants or alleles are actually present. Now, what we really have in our DNA is not just one allele. We have 2 alleles at each location, because each cell has a copy from your mother and a copy from your father. That pair of alleles is called a genotype. And the genotype is what we're trying to identify. In the same way that if you have a building, you're trying to identify the building is made of bricks. But we don't care about the bricks, we care about the building. So the scientists don't really care about the alleles, they care about the genotypes. Because each different genotype represents someone's identity at a particular location. The genotypes are pairs of alleles, one from someone's mother, here there's 10 repeats. Another one from someone's father, say 12 repeats. That genotype would be 10 and 12. And we don't know when we measure in the experiments whether it's a 10 and a 12 or a 12 and a 10. The order doesn't make any difference. Just call it 10 and 12. So many reasons why genotypes are important. The first is if there's about 15 different alleles, different genetic variants at one location, there are about 100 pairs of alleles, just counting them up. And that's a lot of variation. If scientists are measuring, say, 15 different locations, different loci, all across the chromosomes, then there's 100 possibilities from the first location, times 100 from the second independent location, times another 100, and 100 times 100 times 100 times 100. 15 times is a very big number, like a 1 followed by 30 zeros. So there's a lot of these genetic barcodes, these genotypes, when you look at 15 locations. And there's only 7 billion people on Earth, which is less than a 1 followed by 10 zeros. It's a lot more of these genotypes than there are people. And so these become the basis of identifying people because there's just so many more genotypes than people. The other reason they're important is that each cell, human cell in this nucleus, will have 1 copy from the father and 1 from the mother. So you have equal doses. Each cell will have a 10 and a 12 if someone is a genotype at that location on that chromosome. And that means the amount of the alleles is roughly the same. It doesn't have to be identical. It isn't with a lot of variation. but it gives scientists or computers a clue that if you have data that shows a lot of one allele pair at one height and a lot of another allele pair at a different height, then those genotypes of those alleles probably go together. So it's what helps computers or people separate out the genotypes. This is simple DNA evidence interpretation when there is one person who left their DNA. So this DNA molecule indicates it's just one unknown person. And you've heard a lot in previous weeks about what the laboratories do. With the lab, there are many steps that they can do, but they all do three essential steps. First, they extract the DNA from a biological sample to just be looking at the DNA. Secondly, they amplify the DNA so that each copy of DNA turns into millions or billions of copies that can be detected. And the third step is detecting that amplified DNA. That's what the lab does. And when they're finished, during the copying step of amplifying it, they put a fluorescent label on it so it can be detected, and they put it into a machine, a genetic analyzer machine, that separates the DNA molecules based on how long they are, based on their length. The usual picture is the molecules that are shorter are to the left, the ones that are longer are to the right. And if we put in someone's DNA and just looked at one location that was tested, these machines and kits of chemistry are set up that you can test 15 or 20 locations at once and then separate out the signals, so you only have to look at one location at a time, when they're all present. If it was only a 10 and a 12, then ideally you wouldn't see any signal from 8 repeats or 9 repeats, but the allele 10 from a person's parent would have some signal, because there'd be billions of copied molecules that could be detected. We wouldn't see an 11, and then from the other parent, we'd see a lot of signal from the amplified 12 allele. So if the genotype is a 10 and a 12, ideally we'd see signal or a peak at 10, signal or a peak at 12, and nothing else. The data's not that perfect, but it's almost that good with single source DNA. And then inferring what the genotype is out of 100 possibilities. Is this an 8-9? Is it a 7-15? Is it 11-14? What we're seeing in the data is just a 10 and a 12. And so all of those 100 possibilities are collapsed by the data into one main possibility, a 10 and a 12. That's what data does. It reduces choices with different degrees of probability. So about 100% of the probability would go to that 10-12, and that can be done without looking at anybody else's known reference. It's just inferred from the data. And afterwards, a comparison can be made to say, well, yes, it looks like there's a match, and then calculate a match statistic. In this case, it would be about 100% chance that there's a match divided by how rare the 10-12 genotype is in the human population. Say it's 5%, then 100% match, this in the case of the old probabilities, just a match, divided by 5% would give you a match statistic of 20. That would be the match statistic of one location, they'd be multiplied together. But most DNA evidence is not single source DNA. And so we don't have definite answers. The data still reduces choices, but now there will typically be more than one possibility that can explain the data. So here, the two different colors, the two different DNAs, represent DNA from two unknown people. The lab process is the same. The lab will take this biological sample, they'll extract the DNA, they'll amplify it, and then they'll read it out by size at each of the 15 locations. They'll get a signal. But now the signal, shown in the center, doesn't have just two alleles of the same height. Instead, we're seeing 3 data peaks of different heights. This indicates there's a mixture. And then the task of how an answer is inferred, or how a genotype is extracted or separated from this data, is the task of saying, well, so what are the two genotypes that this data is trying to tell us about? So suppose that we know, or we assume, that the first genotype is the 1012 we just saw. Then we ask, what would be reasonable genotypes for the second person to separate out that second genotype? Well, what's new is there's an 11. You didn't see an 11 before. So that new genotype should contain an 11. It might contain a 10 and 11, because we see a 10 and 11 in the data, an 11, 11, or an 11 and a 12. And those three possibilities of 10, 11, 11, 11, 11, 12 are listed. It's possible, but much less likely, that it's an 8 and an 11, because we don't see any data supporting an 8. It can still be there, and just not visible in the data. So those would be much lower possibility events. They wouldn't be up in the highest 99% range. So what we've still done is the data has restricted the possibilities. Out of the 100 possibilities we started with, and assuming that there's equal amounts of the 10, 12 from one person, and a second unknown in the mixture, that second unknown's genotype is a listing of the possibilities, the allele pairs, together with the probabilities. And the probabilities here are reflecting how well combining the genotypes from the two people explain the data. So if the first person's 10, 12, we've got one dose of a 10, one dose of a 12, and we look at the last possibility of an 11, 12, then you have one dose of an 11, a second dose of the 12, that would produce a peak pattern of one dose, two dose. So that would explain the data better than other explanations that might give it a higher probability of numbers that add up to 100%. This is Bayesian reasoning, so there are two parts to it. One is, what are the different occurrences of these genotypes in the population? And the second is, how well does each explanation explain the data? So both have to be accounted for. When it's done, the 100 allele pair possibilities are reduced to roughly 3 with probabilities, and then a comparison can be made to anybody's genotype. In this example, comparisons made to a 11 and a 12. So you see an 11, 12 on the list. The math is exactly the same. The statistic is exactly the same as for single source DNA. But instead of being 100% over 5%, which gave us 20, now it's 50%, because that's what's on the list. It's 50%. 50% divided by 5%. It's a chance of a random person having an 11, 12. The match statistic has reduced by half. It's not 100 over 5, it's 50 over 5. So the effect of the uncertainty is to lower the match statistics, here from 20 to 10. But otherwise, it's all the same. Okay? The lab process is the same. The inference is a little more sophisticated, but you're still ending up with a list of possibilities and making comparisons. This is what the data looks like at one location. This location is from chromosome 13. It's just some spot along that volume. The horizontal, or x-axis, is showing the length of the DNA paragraph. That full paragraph, taking me out to the ball game all the way to the end. The numbers on top are showing how many times that word, in the example is root, is repeated. Whether it's 8 times or 9 times, 11 times. That's the allele, the genetic variant. So horizontally, what we're seeing are the different genetic variants. 8, 9, 11, 12, 13. There are, what are labeled here are 5 different genetic types or alleles. The heights are in relative fluorescence units. How much signal did the machine detect from that allele? More signal means more allele was amplified and detected. A lower signal here 12 as opposed to 13 means that less of that allele was amplified. So that's what data looks like. And computers can do a lot with this data. They can use the heights of the peaks. They can use the pattern they're trying to explain, like medium, high, low, high. They can also use the variation between the peak heights to calculate probabilities. So a lot of information here. People have a very different way of interpreting data. It's hard for people to solve a 100-dimensional probability set of math equations. Actually, they just can't do it. It's not that it's hard. It's like actually basically impossible. Even if they had the math written out, it would take them maybe 10,000 years to go through a process of doing all the calculations. So they're not computers. So what computers do instead is what people do instead is they simplify the data so that people can actually understand it. And the way they simplify it, and have done for 20 years in a simple form of human review, is they get rid of the pattern of the high peaks, the low peaks, the variation. They draw a line called a threshold, and they just say, are these different peaks in or out? So here, 9, 11, and 13 are in, and everything else is out. How much that's present isn't used. The peaks underneath the threshold, they're not used. And instead, all of the data in this pattern are reduced to the list 9, 11, and 13. It has a lot less information in it. Also, if the threshold is adjusted up or down, there might be peaks that could be in or out and could change the answer. That's what people were doing by hand 20 years ago, and many labs still do it, but most have or are moving on to using computers. And this is how the computer solves a problem. It tries to explain the data. Bayes' reasoning has two parts. How prevalent are the different genotypes as explanations, and how well does adding together the genotypes from the different contributors explain the data. So we'll focus on how well the data are explained. It's called the likelihood part. So the data are not going to change. An important part of modern statistical reasoning is that we don't change the data. We don't say, well, maybe peaks at a height of 100 or so far above the noise that we'll use those, but not things that are lower that might also be real data. Instead, all the data are used. And the task is to explain the data. In a complex mixture, Trulli will usually try out 100,000 different explanations. Every cycle of this 100,000 cycle process, the computer will try out an explanation for the data at every one of the 15 locations and for another 50 variables for how much of each genotype is present, what the variation is, artifacts that we won't go into like stutter, shadow band, which are shadow bands. The computer uses math to propose different solutions, and every cycle tries out all these different variables and sees how well they explain the data. So this is what an explanation looks like, simplified just to adding together genotypes from 3 people. So the computer might propose there's a first genotype, and there's a lot of that genotype because the bars are taller. There are 2 bars because it's a genotype is a pair of alleles. The height's telling us how much, the color will tell us which genotype, and the actual alleles are telling us the value of the genotype. This is an 1113. So suppose that first the computer says 1113, that's the first contributor, shown in blue. And then it proposes and adds a second contributor, shown here in what looks like brown, though it's supposed to be a different color, but just computers. So here's an 8-9 allele pair. That's proposed as a second possible person's genotype. It's about half the height of the first person. So the computer's saying there's less DNA from the second contributor, shown in the second color. And then it proposes a third contributor, shown here in green, with a little bit less DNA still, and that's a 9 and a 12. And then adding together the amounts of alleles from all these three genotypes produces a pattern. And the pattern we see is medium, high, high, low, high. It's the same pattern that we saw with the data. So this explains the data pretty well. It's producing the same pattern. What the computer is actually doing inside, aside from a lot of probability math and so on, but first level, it's looking at what's the height of the added together genotypes, the top of that stack, say at allele 9, with the first allele from the second person and another allele from the third person. That height is around maybe 160 RFU. It compares it with the data peak that's underneath it, which is also at around 160. It's a very small deviation. So when the deviations are small, it just subtracts the numbers, squares them, adds them up. When the deviations are small between what it's trying to explain in colored rectangles and the data that isn't going to change underneath it, that's a good explanation. It has a high likelihood. Most proposed explanations are terrible because there's so many possibilities. And when it tries out an explanation like the one here, where the genotypes are not really explaining the data, those genotypes, those allele pairs for each of the contributors get a lower probability. And the computer tries out many, many possibilities. And that's how it can work out the probabilities. When it's done, for each of the three contributors at each of the, I think it's going to be about 15 locations, there will be a genotype for one location and one contributor. So 3 locations, 15 genotypes would be 15 locations would be 45 genotypes. This is one of those 45 genotypes for this D13 location for that first blue contributor. And this is what the genotype looks like after having seen the data, applying Bayes' theorem. There are 100 possibilities that would go from that wall probably around the courtroom. But these are the 10 where most of the probability has been concentrated. That's what the data does. It concentrates the probability. And what we're seeing horizontally is a listing of each of those possibilities. Here's the 8-9 genotype allele pair, an 8-11, a 9-11. They're listed. And the other 90 aren't shown because their probabilities have gone down. And vertically, we're looking at how much probability there is. So in probability, the numbers will add up to 100%. They don't go below, they don't actually go down to 0. They can be very, very small. But for the 100 possibilities, probability has been reapportioned to give this distribution of some probabilities rising and some going down. This is what we started with. This is the random population genotype. This is what we'd expect to see if we just went downstairs in the courtroom, swabbed somebody's genotype, and we asked at this one location, out of 15, what's the chance they would have any of these, say, 10 genotypes. The computer will consider all 100 of them, but these are the 10 that are shown. So these are the same 10 that we just saw, and some genotypes, like 11, 12, are prevalent in a quarter of the population. Others are much rarer, like the 8-11 is only in 1% of the population. This is what we started with, and after looking at the data on the previous slide, that's what the computer ended with. That was the data changed the probabilities from brown coincidence to blue evidence. And here they're shown together, same bars. And now a comparison can be made against any of the 100 possible genotypes, 10 are shown here, to ask what is the change in probability? What's the ratio of where we ended up after seeing evidence in blue divided by coincidence, the chance that this just could have happened randomly. And almost anything can happen. If we take a look, 3 from the right, we see at 1113. If this actually was an 1113, the chance of a coincidence started as the height of the brown bar. It roughly doubled after seeing the evidence. So if we compared this genotype, this listing of possibilities and their probabilities, with an individual whose genotype happened to be 1113, that ratio would be 2 and would be a bit of evidence that might point towards somebody having left their DNA. On the other hand, if we compared it with someone who had a 1213, the chance of coincidence is twice the chance of the evidence. So after having seen the data, that number divided by the chance of coincidence is a half. The blue bar is half the height, having seen the evidence divided by coincidence before we saw the evidence. A number like a half would point away from someone who had a 1213 genotype after the computer looked at the evidence. If the bars were about the same height, that would be a ratio of 1. That wouldn't tell us anything. That would say the data didn't have an impact either way. In this case, we're making a comparison with an individual whose genotype is an 1112. Could be anyone, but this is where we'll focus. And we see that before looking at the evidence, the chance that somebody had an 1112 genotype was 25%. After looking at the data, the chance was 10%. And so 10%, the chance after seeing data, divided by 25%, the chance of a coincidence, is 2/5, or 0.4. That's less than 1, and that would point away at this one genetic test of a person having an 1112. The same process is done at all 15 tested locations. They're all tested on the same sample in the same tube, and they're amplified together from different independent locations on different chromosomes across the human genome. And this is the result. We just saw that number that was less than, oops, no. This is an unusual clicker. It's like a very, very tiny button. Getting very low margin of error. Okay. So here are 15 locations. This is 1. 1 means there's no information either way. Some locations, the number is bigger than 1. Other locations, the number is less than 1. The locations are listed here. There are 15 of them. And that's the strength of the match. These are independent locations. And so statistically, we can multiply these 15 numbers together, some bigger than 1, some less than 1, to get the final match statistic. What do all 15 tests tell us? And in this comparison, we can look at this one, at all the 15 locations from the evidence that corresponds to the right bra cup, as it was compared with the reference of Charles Merritt, and compared to different ethnic populations. That's the brown number on the bottom. The coincidence, we arrive at different match statistics. These are the populations that were studied by the FBI, informing the chance of a coincidence. Just looking at the smallest number, without even regard to someone's ethnicity, the reason the different ethnic groups are used, I think the field may be moving on to just using one composite database. This is how it's done. It's been done for 20 years. So it's safe to say if we're not going to worry about ethnicity, just use the smallest number if you want to be conservative. And that's what's pretty standard in the field. We could just look at the smallest number. I think it's the one on top, 3 million. And then just reading off the formula in English in terms like match and probability, we can say that a match between the right bra cup and Charles Merritt is 3 million times less probable than a coincidence. Another way of saying it is that it's a match between the right Brock and Charles Merritt is 1 in 3 million times more probable than coincidence. Well, whatever it is, coincidence is 3 million times more probable than it being this person. So it's evidence that points away from this person. And then to answer the question, because we looked at many items of evidence, and we looked at many references, are these references in the evidence? When we compared about a half dozen evidence items with 5 references, we can say that based on the Truller results, comparing all of the evidence genotypes with all of the references, they all produce exclusionary match statistics, numbers that are less than 1, which can be expressed as 1,000 times less probable or 1 in 1,000 times more probable. It means the same thing. It's a number less than 1. And so I'll just go through what we found for the different items of evidence.
Actually, let me interrupt you for a minute. It's just about 12, so we'll go ahead and take our noon recess. This time until 1:30. Come back at 1:30 and pick up. Okay. Again, keep in mind the admonitions previously given to you not to form or express any opinions about the case, not to discuss the case, and we'll see everyone back at 1:30.